Okay, we are online, so I will go ahead and get started. Hello and good morning, everyone. Welcome back to our Ask a Scientist segment with the Asheville Museum of Science. My name is Abby, and I'm one of the educators with the museum. And today we have with us Josh Manning, who's working on completing his PhD at Florida State University. Again, he's where I wish I was, just in that nice, warm Florida State. Josh studies coral reef ecology and looks at Caribbean parrotfish and how they play a role in keeping reefs healthy and whole functioning ecosystems. So feel free to send in questions along the way for Josh, and we will get started. He's got lots of cool pictures to show us from some of his really cool dives. So this should be a really interesting talk. And Josh, you can just go ahead and take it away.
All right, cool. Thank you. I'll go ahead and share the screen here and make sure everything's going. There we go. All right, can everybody see that? Good? Awesome. All right, so my name is Josh Manning. I am a PhD candidate in Dr. Sophie McCoy's lab at Florida State University. And I figured, I might start this talk a little bit talking about how I got into marine science to begin with and then get into talking about my current research. So here, let me pull this up real quick. All right. So my interest in the marine sciences kind of began when I was growing up on the Gulf Coast of Florida when I was younger. I was always interested in the ocean. I loved animals in the ocean. But it wasn't really until I moved to Maine that I started to get involved in marine science. And that began when I was in high school. And during high school, I volunteered for community service at a local aquaculture facility at the University of Maine. And at this aquaculture facility, we did a lot of breeding of ornamental aquarium fish like clownfish and dottybacks, like the ones shown up here. And so this kind of was like my first introduction to coral reef sciences as well. So during this time breeding these, I was learning a lot about destructive fishing practices and the reasons why we were aquaculturing these fish. And that got me really interested in coral reef ecology, but I still had to get into college, and I didn't really know what I wanted to do yet, but I figured, marine science sounded like a good idea. So I applied to the University of Maine to join their marine science program. And when I got there, I started to volunteer in other labs and gain more interests in different aspects of marine ecology, including algal ecology. So a lot of the time that I spent during my undergraduate was focused on studying algae. And this was done in the intertidal zones, but I also studied unicellular algae for an NSF research experience that I had in Texas for a summer. And I studied these dinoflagellates that cause harmful algal blooms and looked at how different things affected their growth. But the real turning point for me was during my senior year of college when I got to work with Dr. Bob Stenick, who is a coral reef ecologist who's done a lot of work around the world. And he focuses his work on coral reefs, but he also studies algae. So it's kind of like the sweet median between everything that I'd learned up to that point and then transitioning into coral reef ecology. And it was with him that I first got to go to a coral reef. And I did this in Bonaire, which is, pretty cool because now all of my research is in Bonaire. And Bonaire is this island in the Southern Caribbean, and I'll talk a little bit more about that later. But this really got me going on this coral reef track. But before I could continue on that, I graduated and I took a year in between, and I spent some time doing marine education at a summer camp in Seattle, or outside of Seattle, and just taking kids around the intertidal zone and showing them, all the different animals, trying to get them excited about marine science. And after that summer, I returned to Maine and I decided to work in aquaculture a little bit more. And this time I worked at the Center for Cooperative Aquaculture Research. And at this aquaculture facility, we weren't focused on small ornamental reef fish. We were focused on large food fish. So cod, these yellowtail amberjack here, which are a big sushi fish, and then halibut. And so I spent a lot of time taking care of the fish, doing animal husbandry work, feeding them, cleaning their enclosures, things like that. And, this was really fun. And I actually really enjoyed doing this, but it wasn't quite what I wanted to do. And I knew I wanted to go back to school and pursue the coral reef ecology. So that's what I did. And so during that time, I began writing up fellowships and things like that and looking for grad schools. And I found a position with Dr. Bob Carpenter at California State University in Northridge, which is in LA. And this was like the best thing ever for me, because I got to go to Moorea, which is an island just off the coast of Tahiti. That's a ferry ride away. And I spent 2 years there every summer for a few months just doing research. And my research at this point focused on coral and algae. So this was focused on these calcified algae shown here. And these calcified algae are a framework builder on coral reefs. And they don't get a lot of credit, but they're actually really important in consolidating the reef substrate. And my research during this time focused on how ocean acidification and climate change were affecting growth and calcification of these important calcifying algae. And one of my major interests was in understanding how ocean acidification affected regeneration from wounding. So coral and algae are really important framework builders, but they also have to deal with a lot of grazing by urchins and fish that leave a lot of scarring in the surface of their tissue, which they're able to regenerate under normal circumstances, but I wanted to know how they were affected by climate change. So I did a lot of experiments where I spent a lot of time in lab doing water chemistry and water quality analyses and things like that, looking at how ocean acidification was affecting them, specifically their calcification, but also regeneration of wounds. So I created wounds in the surface of these algae, and I placed them into the treatments, and then afterwards I cross-sectioned them to look at how much tissue they had regenerated. And I did this using scanning electron microscopy. So this is a scanning electron microscope image of the thallus of a coralline alga. And you can see all these little holes here. These are all cells. So I'm looking at the cellular level at how much regeneration had occurred within these wounds that I made. And what I found was that ocean acidification is actually reducing their ability to heal these wounds, which has implications for their ability to compete with other species of coralline algae, and also to generate the framework that they create on reefs. So I really enjoyed this work, and I loved spending time in French Polynesia. Still one of my favorite places I've ever been. But it was a lot of lab work, and I kind of wanted to do more field work. So I wanted to be in the water more, interacting with organisms and kind of getting to understand their ecology. And so I began applying for PhD programs, and during that time I found Dr. Sophie McCoy, who I think gave a talk a couple weeks ago for this program. And she studied a lot of the same things that I was studying, but in temperate systems and in intertidal zones. And I spoke with her, and I really enjoyed talking to her, and I really wanted to join her lab. So I applied, and she took me on. And my initial plan was to study coralline algae still, and look at how they healed wounds in the reef itself. So, actual natural wounds created by herbivores, and look at how they were regenerating the tissue, and look at this at a cellular level. But the first year that we went down, me and my lab mate went down to Bonaire, we did a lot of diving. And my observations during that time frame, all my natural history observations, actually led me to a completely different question. And I'm going to talk about that today a little bit. But I definitely spend a lot more time in the water now, sometimes 300 hours in a single summer. And it's a lot of fun. And I'll get back to the fun part of this afterwards, although I think the research is fun too. So my current research now focuses on the spatial ecology of Caribbean parrotfishes. So I'll talk to you about the importance of parrotfishes to the coral reef structure itself, but also about their behavior and how it shapes where they are on the reef. But first, I'm going to introduce, general coral reef, what coral reefs are and what stressors they're facing, and why this is important. So coral reefs are ecologically and economically important. Their diverse communities support the livelihoods of millions of people around the world. Their structure that they create provides coastal protection from storms and damaging waves. But coral reefs are increasingly degraded by human impacts. So overfishing, eutrophication, climate change, these are all big ones that are affecting coral reefs today and leading to the loss of coral in these systems. So we're transitioning from healthy coral reefs that are dominated by corals, like the photos you see on the left, which support a large diversity of fishes and other organisms, to more degraded states that are dominated by, in some cases, fleshy algae, but in other cases, cyanobacteria. And this is an example here in the bottom right of a cyanobacterial bloom in Bonaire, near one of my study sites that was approximately 1 kilometer long. And this cyanobacteria was smothering entire beds of this endangered Acropora. So these human stressors are leading to a lot of shifts in coral reef benthic communities, but primarily to ones dominated by autotrophs like algae and cyanobacteria. And herbivores can help to prevent these shifts. So herbivores directly remove algal biomass from the reef substrate. And on the right, this is a plot from a paper by Williams and Flunen in 2001, where they show the effect of herbivorous fish biomass on the percent cover of macroalgae on the substrate. And what they found was that when they looked at a bunch of different sites, they found that the abundance of fish, herbivorous fish, actually reduced the amount of macroalgae on the reef. So as you increase the abundance of these herbivorous fish, the amount of macroalgae also decreased on these reefs. And this removal of algae can have indirect effects on the benthic community by reducing coral algal competition. So algae is a main competitor for space with corals on coral reefs. And by removing that, it allows corals to grow more freely. And it also increases the available settlement, habitat for settlement recruitment of coral larvae. So the baby corals like to settle where there isn't a lot of algae, and preferably will settle on crustose coralline algae in some cases. So by removing the fleshy macroalgae and promoting an early successional state dominated by coralline algae, herbivores are important in promoting coral recruitment. And parrotfishes are considered some of the most important herbivores, particularly in the Caribbean. And they're the different parrotfish species target different things. So here on the right, I have a plot of the proportion of bites taken by 9 different species of Caribbean parrotfishes. And you can see basically in this plot that they target a lot of different things, and that some species target some things more than others. So some species target more macroalgae, as is the case here. All this red here is macroalgae. While the species over here on the right, mostly scarce species with the exception of one Sphaerosoma species, target a lot of turf and crustose coralline algae. So they're targeting different things, but they are removing algal biomass during foraging. But despite the fact that macroscopic foraging observations have shown that they are removing algae, recent nutritional evidence and evidence from their digestive anatomy, including the pharyngeal jaws and things like that, and their ability to digest different materials, suggests that they're actually targeting cyanobacteria more so than the algae itself, and that they're removing the algae as a byproduct of the grazing. So they're targeting the cyanobacteria that grows on and within the reef substrate, and they're gaining a lot of nutritional value from this. And recent work by my lab mate and I and my advisor found that one species of Caribbean parrotfish, Scarus iseri, targets benthic cyanobacteria in high proportions. So here we have a plot of the proportion of bites again for this species, the striped parrotfish. And you can see this red here. This is all benthic cyanobacterial mat. And these benthic cyanobacterial mats are just blooms of cyanobacteria on the reef substrate. And so we found that they're targeting these pretty frequently, which supports the idea that they're getting a lot of nutritional value from cyanobacteria. And I found this again when I studied 5 of the most common species of parrotfishes on these reefs. They're targeting these benthic cyanobacterial mats and with preference. So 4 of the 5 species were targeting this preferentially. And you can see an example of this here, this red-orange mat here. This is all cyanobacteria, and this is a princess parrotfish targeting that cyanobacteria. And this may be an important mechanism in reducing or controlling cyanobacterial blooms. So like the one that I showed previously on the reefs in Bonaire. But regardless of what they're targeting for nutritional values, they are removing algae and cyanobacteria, which creates space for coral larval settlement and also reduces the amount of competition with corals. But parrotfishes are also corallivores, so they will sometimes eat corals. And different species will target different species of corals. This is work that I recently did, suggested that they like different species. So different species of parrotfish will target different species of corals. And this can cause extensive damage to the coral tissue. And this is an example here of a stoplight parrotfish targeting a live coral. And you can see that it creates a lot of damage to the coral tissue. And this can affect their physiology, reducing growth rates and reducing reproduction. But it can also affect their distribution on the reef, depending on the frequency and intensity of parrotfish grazing in different locations. But luckily, corallivery seems to be less important than their role as functional herbivores, and in general, they have a positive influence on the benthic community. So another thing that could complicate this a little bit is that parrotfishes have complex social and mating systems. So parrotfishes are protogynous hermaphrodites. They transition from female to male at a given size class. So they transition from these more drab initial phase females to these colorful, larger terminal phase males. And territoriality is common in these social systems where terminal phase males, like this one here, will defend harems of females. And they do this because it increases access to mates and resources. So here we have an example of a terminal phase stoplight parrotfish mating with an initial phase parrotfish. So they do these really cool spirals up into the water column where they'll meet up and they'll swim up into the water column, and they'll do a quick release of gametes, and then those eggs are fertilized in the water column and eventually will settle back onto the reef as juveniles. And so this behavior, this mating, is more common when the males are maintaining territories. But territoriality could also constrain movement and therefore foraging, and it could contribute to spatial heterogeneity in their effects on the benthic community. So it could dictate where they're grazing and where they're removing algae and promoting coral recruitment. So my main question for my research currently is how does territoriality mediate parrotfish space use and foraging, and what are the top-down effects on coral reefs? So for what I'm going to talk about today, I'm going to focus mainly on one of my chapters, which was quantifying the spatiotemporal patterns of parrotfish space use and the role of territorial behavior in driving these patterns. So my main hypotheses for this portion were that home range size would differ by species and ontogenetic phase, so between initial and terminal phase. And this is because body size is known to correlate with home range size. So larger species, larger individuals are known to have larger home ranges than smaller species and individuals. I also predicted that territorial behavior would result in limited spatial overlap of the home ranges. Therefore, these home ranges might actually correspond to defended territories, and that the territories would remain temporally stable over time. And there's a little bit of evidence for this in the literature. And this is an important thing if territorial behavior is going to have an influence on what is growing on the reef itself. So to study this, I work in Bonaire, which is an island in the southern Caribbean. It's about 60 miles off the coast of Venezuela, and it's a part of the Netherlands. And I studied, or I conducted my research at 5 different sites on the leeward side of the island, so on the west side of the island. And I did this at sites that have high coral cover and low macroalgal cover. And this plot on the right basically just shows the percent cover of different benthic organisms at these sites. And you can see that coral cover is relatively high. And in general, coral cover in Bonaire is higher than most places in the Caribbean. They still have really, really healthy coral reefs, despite a lot of disturbances, including bleaching events. And at these 5 sites, 5 species of parrotfishes make up about 96% of the biomass of parrotfish. And these 5 parrotfishes are shown here. So we have the queen parrotfish, princess parrotfish, stoplight parrotfish, red band parrotfish, and striped parrotfish. And at each of my 5 sites, or across my 5 sites, I tracked several terminal phase males of each of these species, and also several initial phase females of these 3 species here. And these tracks look something like this. So I'm underwater conducting behavioral observations. So I'm following the fish from a distance, recording its behavior on a GoPro, while another snorkeler, in this case my advisor, snorkeled above the fish, tracking its locations with a handheld GPS unit. And these GPS units are basically like the Garmin's that you would have in your car. And they take accurate relocations of the fish. So they collect GPS data and store it for me so that I can analyze it later. And this is what these tracks look like. So each of these different colors on each of these plots is a different individual. The different points represent different relocations. So that's where we got a GPS location for the fish. And you can see that for these 3 species here at one of my sites, that the movements are kind of restricted, and they're by themselves most of the time. They're not overlapping with the movement of their neighbors, which is pretty cool, right? That indicates that maybe these are actually territories. And if you overlaid these, you'd see that all of these species are occupying the same exact spaces on the reef, which indicates that they have partitioned their feeding enough that they can coexist in the same locations, or partition their resources in some way that allows them to coexist in these areas. But this isn't the traditional way to analyze these kinds of data. So traditionally what we would do is we'd look at the home ranges. And so I took these individuals that I showed before, and I've now presented the home ranges for those same individuals. And basically what a home range is it's a representation of the probability of finding an individual in a given space. So you use the relocations that I showed before, and you create a probability distribution of where you might find an animal. And then you can plot the area that you might find it 95% of the time. And that's what these are here. So these are representing the home ranges for each individual of these 3 different species. And you can see that still there's relatively little overlap between most of these home ranges, suggesting that they are territories. So the next thing I wanted to do was look at whether or not there were differences between species and phase. So I plotted the home range area for each species, each of my 5 species plotted by phase here. So initial phase on the left, terminal phase on the right. And what we found was that these smaller species had smaller home ranges. So Scarus tanioperus, which is a small scarus species, had smaller home ranges than all of these other species. And Sparosoma arfinatum had smaller home ranges than the other Sparosoma species, Sparosoma viridae. And we also found that initial phase, these small females had smaller home ranges than the larger terminal phase males. So again, this is likely a body size effect. So as you increase in body size, you need more resources, and therefore your home range is going to expand to allow you to get all of those resources. And so the next thing we wanted to know is whether or not these home ranges were overlapping and whether or not they correspond to territories. So to look at this, we computed an overlap matrix that ranges from 0 to 1, 0 being no overlap, 1 being complete, perfect overlap, and they're in the same place. And what we found was that for pairs of the same species, so conspecific pairs, overlap was really, really low. And it was lower than it was for pairs of different species, or these heterospecific pairs. And this is likely because of the proportion of interactions with conspecific. So if you plot the proportion of interactions for each of these 5 species with other parrotfishes, you find that they're interacting with individuals of their own species more than individuals of other species. And these interactions correspond to posturing displays. So up here in the left, this is an individual of terminal phase Scarus vetula, and it's posturing, flaring its fins out, making itself look large, and it's squaring off with a neighboring individual. But they can also be more aggressive, as is the case here in the top right. So these are 2 neighboring terminal phase stoplight parrotfish, and they're chasing this non-territorial floater male through the boundary of their territory. So often when we'd follow these fish, we would notice that some individuals didn't have territories, and these individuals would bounce from territory to territory, getting chased off every time. So they readily defend these territories from conspecifics, especially ones that don't have territories and are trying to move in. So this indicates that these home ranges actually represent or correspond to defended territories. So the next thing that we wanted to know is whether or not these territories are stable over time. Do they stay in the same place? And if they do, what effect might this have on the benthic community? So to determine whether or not they're stable through time, we repeated tracks of the same individuals at different time points. So about a month apart during the summer. But I also have, for Sparosoma viridae here, this green cutout outline. I tracked individuals in January of 2019 and again in the summer of 2019, about 5 months apart at 2 of my sites. And we use the same overlap metric to look at how stable they are. So higher overlap would indicate that these were stable and in the same locations for a longer period of time. So what we found was that there is actually really high overlap. So they're in 75% overlap in most cases, indicating that these are relatively stable through time, between 1 and 5 months, which is really important because if this behavior is going to be affecting where they're grazing, you would need it to be for a long period of time for it to have an effect on the benthic community. So in conclusion, what we found was that smaller species have smaller home ranges, smaller individuals, so smaller initial phase individuals have smaller home ranges than terminal phase individuals, again, supporting prior work that has found that home ranges are correlated to body size. These home ranges actually correspond to territories. They were heavily defended, generally against conspecifics, so individuals of the same species, and there was little overlap between them, indicating territories. And these territories were stable through time at scales of 1 to 5 months, which is really cool. So what does this mean for the benthic community? So my interest is in understanding how this territorial behavior affects what's going on in the benthic community. So what things are present in that community. And so we can imagine that if if two terminal phase individuals are maintaining territories next to each other, and they're spending a lot of time patrolling the boundaries of their territories to defend it from other individuals and make sure that they can maintain that territory, they may not be grazing a lot in these areas. And they may be focusing their grazing in the center of their territories. And this may lead to differences in the communities that are able to establish in the benthic community. So if you're grazing a lot in the center, that might reduce the algal cover and promote coral recruitment. And therefore, we might see more corals in the center of the territories than the edges. And again, this depends on the amount of time that these territories are there and consistent. And we've shown that they are. So my future work over the next year is to determine these effects. So I'm conducting experiments, and I'm going to be looking at how the community changes within these territories, depending on how grazing is distributed within the territories. So that's going to be the focus of my future work. And at this point, I would like to thank everyone who's helped out with this. This is a big group effort. So I had a lot of undergraduate and summer interns that helped out with this work, and they came down for the whole time, and they were in the water with me most of the time doing this work with me, and helped to analyze it after the fact. We also have local connections that help to get permits to work in the National Marine Park there. Bonaire's reefs are some of the most protected in the Caribbean, and it is difficult to work there because of these protections, but it's for the best because they still have really healthy coral reefs. And I'd like to finish up by also talking a little bit about why I love diving to begin with, and why I love research to begin with. And diving is more than a tool for research. It's also a lot of fun. I do a lot of following of parrotfish and doing all these things, but I often I spend some time underwater looking for other things as well. And often I'll find really small things if I'm looking carefully. So all of these things, with the exception of the angelfish here, are really small, about the size of my thumb. So this frogfish, watching it walk around on the reef is really cool. So they use their modified fins to walk around, and they have a lure on their head that they use to attract prey, and they ambush them. And they're really hard to find because they're super well camouflaged, but that was maybe one of the coolest things I've ever found. Octopus, little tiny fish, they're super colorful if you can see them quick enough. And we also see a lot of really big, cool things as well. So I figured I'd share some of these videos. This is a barracuda that I found under a pier as I was leaving one of my sites. It was probably about 3 or 4 feet long. It was a pretty big fish. Super cool. Definitely made me a little nervous at one point there. But generally awesome. Eagle rays, we see these all the time. This is a resident eagle ray at one of my sites. So pretty much every dive I did at this particular site while I was doing research, when I would come back in, I'd see this eagle ray. And he's sitting in there right now, foraging for food. So, digging into the sediment, trying to find crustaceans and things like that to eat. And it let me this day, for some reason, get really, really close to it. So I was able to get a really cool video. Sometimes we get surprise visitors. So this was the last dive of my first summer in Bonaire. With me and my dive buddy, we're getting ready to do transects to measure the abundance of corals and things like that on the reef. And this Manta Ray probably had a 10-foot wingspan. and it swam right over us and just kept swimming down the reef. So we get to see really cool things like this sometimes. This is pretty rare occurrence. I think we've seen 3 in our entire time in Bonaire, and each of them actually on the last days of our diving. So it's a Bonaire send-off. And then occasionally we see sea turtles as well. They do a really good job of protecting their sea turtles in Bonaire. They have nest locations. You can even track sea turtles. So I bought a hat from one of the dive shops, and they had this program where you buy a hat and you get a tracking number, and you can track one of these sea turtles through its lifetime. They attach trackers to them to monitor their health and things like that. So we see a lot of other cool things as well. It's not just research, but be a lot of fun. So I'll take some questions if anybody has questions and would like me to go back to anything.
Yeah, those videos are awesome. Have you ever, besides the Barracuda, have you ever had a moment underwater where you've been afraid of something?
Not really. I mean, honestly, the Barracuda, it was definitely like, it looked like it might have jumped at me a little bit there, but I probably scared it more than it scared me. No, I haven't really been all that nervous underwater. I've been in the water with some pretty big fish, including sharks. I've seen my fair share of sharks during diving. I think maybe one of the most startling experiences, mostly because I had like no visibility. I was diving off the coast here in Tallahassee, out in the Gulf Coast, and visibility was maybe 15 feet this dive, and we were down there. Some people started fishing from boats above us, and we were on this offshore reef about 13 miles out, and this big 10-foot great hammerhead swam by, and I didn't see it until it was like right in front of us. So that was a little startling, but it swam away as soon as it saw us. It was, again, probably more afraid of us than we were of it.
So can you kind of compare some of the diving that you've done in Maine and in the northern coast to more of the southern areas that you've done?
Yeah. So actually, I've been fortunate enough to dive in a lot of places. So I spent some time diving in Seattle as well. So I've done a lot of cold water diving in Maine and Seattle, but also on the coast of California and the kelp forests. And I'd say the biggest difference between diving someplace like Maine and down in the Caribbean, for instance, is the visibility. So it's the cold, but also the visibility. And the visibility is a big thing because it makes it really hard to navigate sometimes. But I did my training dives in Maine, so I was used to really strong currents, really bad visibility, and I think it actually made me a better diver because I was trained in those kinds of conditions, because it made me more aware. I was able to learn to properly navigate in those conditions. And once you can do that, like transferring that to a reef where you can see 300 feet where there's limited current, it's not that bad. So I think that's probably a big thing. But I don't particularly miss diving in the snow and 40-degree weather. I do love diving in the Caribbean and elsewhere in the tropics.
We have a really good question from Ben, who's watching from Vermont. And he said, what is the biggest thing someone from a landlocked state can do to help support the health and well-being of our oceans?
So there are a lot of things you can do. You can, advocate in your daily life, just talk about the issues facing the ocean with people who, like you, are going to have a say in what happens, right? You can change your own personal daily activities, maybe try to use your car less or anything like that. And I think the biggest threat facing coral reefs in particular is climate change, right? And we can all do a little bit to help out with that. And vote. I don't know if you're a voting age, but when it comes time to vote for people who are going to take environmental issues seriously. I think that's the biggest thing you can do.
And Greg asks, what advice would you give to a young person who wants to study sea creatures?
Do it. Get out there and do it. I mean, I don't know. Find opportunities where maybe you can volunteer and get experience working in someplace where somebody studies animals. Spend time in nature. Honestly, one of my favorite things to do when I'm not doing research is to be outside in nature, taking pictures. You learn a lot just by being in nature. So if you're really interested in it, get out there and look at it if you can, and kind of just look for patterns, try to figure out what's causing the patterns, and as you get older, keep pursuing that interest.
Yeah, that was actually something I'm kind of interested in. When you were down there diving and looking at your parrotfish, did you ever notice like odd behaviors that you're not even studying, but it was just these patterns? Like, did they have personalities?
Yeah, I don't know about the personality necessarily, but we would definitely see different things, like weird things that you notice a lot of things when you spend that much time with something. So this whole project was based on natural history observations. My first summer we went down there. I had a completely different agenda. I was planning on studying algae, coralline algae again. I went down to study coralline algae, but in the process of looking at that, I noticed that the parrotfish were swimming in circles basically over the same patches of the reef, and I was like, wow, why are they doing this? And I had no idea like how territorial parrotfish were, and that kind of led me down this path. So it was all observation and just noticing things that, if you don't spend time watching, you wouldn't notice maybe.
Do you have a favorite parrotfish species?
Yeah, actually, and it's not one of the main 5 species that I study. It's the rainbow parrotfish, and I wish I had an image of it. I have some pictures of them. They're really big, so they can get to be 3 feet long, right? About a meter long, and they're really big. But they got those big teeth, like classic parrotfish teeth, dentition, like look like they're smiling, but they've always got algae growing in the side of their mouth, and they take really big bites, and you can hear them coming before you even see them, because they'll be taking bites of the substrate, and you can hear them scraping and removing the substrate.
That's exactly what the next question was. What kind of sounds do you hear when you're diving?
Yeah, a lot. We hear a lot of sounds. In addition to hearing divers and boats and things like that going by, the parrotfish are probably the most noticeable thing. Them taking bites of the reef, you can hear it. They're constantly scraping the reef substrate. And parrotfish, in addition to being herbivores and corallivores, they're important in bioerosion processes. So many people know this now. It's been kind of a popular thing to talk about. Parrotfish poop is actually what creates beach sand. And it is true. So when parrotfish are grazing, they're removing calcium carbonate from the reef substrate, and they grind it up in their pharyngeal mill. And when they poop out basically sediment. It's basically sand, and they distribute that all over the reef. And that's one of their major functions is in bioerosion and sediment production.
And what is your favorite memory of diving?
Honestly, that Manta Ray. I think the Manta Ray might be my favorite thing ever. It was, I don't know, it was a surreal experience. We were laying out this transect. My dive buddy was looking down at his clipboard, and I was looking at him. I was watching him, make sure he didn't need anything. This was like for his work and his research. I was just helping him, and I kind of felt this shadow go over the top of us, and I was like, "What is this?" And I looked up, and this massive Manta Ray was right above us. And he was so focused he didn't notice anything. I was tapping my tank trying to get his attention, and he wasn't looking, and I was just like, he'll figure it out. And I kind of just started following after it, and I looked behind me, and he realized, and he was like, oh, and he was trying to catch up, and we just followed this. And I think the coolest thing about it was like how little effort the Manta Ray actually had to exert to swim through the water. We were going against the current. I was kicking so hard, and I could barely keep up with this thing, and it wasn't even moving. It was just kind of gliding through the water, and it was a really cool, surreal experience.
And then I have one question about kind of going back to the beginning when you mentioned that you were looking at the coral reef cell regeneration. Did you notice that climate change did it affect like the cells, like how they looked? Did they look abnormal at all, or did they just grow back slower?
Yeah, so I didn't look at that in particular, not in any detail at least. There did seem to be maybe some differences in the cell wall thickness. So this is something that my advisor, Dr. Sophie McCoy, has looked at with temperate species of coralline algae. So she was conducting experiments and things like that in intertidal zones in Washington State, and she found that acidification is affecting the morphology of the cells by changing the thickness of the cell walls. So rather than slowing down their growth rates, they're just calcifying less. And there's evidence from other animals too, like mussels and things, that they're changing the structural. So the structure of the calcium carbonate that they're depositing, the structure of the skeleton is changing slightly because of the acidification. And we think it's just a trade-off between growth and like maintaining growth, which is their main way of competing, but also, continuing to have calcified cells.
Do fishermen fish parrotfish typically, or does that not happen?
It is actually a major problem. So overfishing has been a big issue on many Caribbean reefs and reefs around the world. And parrotfish are one of the main targets of traditional artisanal fisheries. So in a lot of Caribbean islands, they've had trap fisheries where they'll target parrotfish that way. And luckily in Bonaire, they've protected parrotfish for a long time now. They put bans in place, so it's a protector, which has led to an increase in their abundance in Bonaire, at least, or maintained abundance. And other places, you can't say the same thing. Like Jamaica's reef fish populations have declined substantially. You don't get large parrotfish there anymore.
And how fast are these populations able to bounce back after that? Assuming the problem's fixed.
Yeah. Well, after the fishing is ended, there is some evidence that they'll increase fairly quickly, but there's kind of a lot of back and forth on that. No real consensus about how well marine protected areas and things like that work. But in general, it does increase the abundance of fish and the biomass of fish on marine protected areas. So it does work.
Yeah, all right. So we don't have any last questions coming in, but if you have any other things that you want to leave us with, that would be great.
I think, if you're interested in marine science, I would say go for it because it's awesome. I love it. Pay attention to the environmental issues. They are important. Climate change is a real big thing. And I think that, that should be the main priority, and hopefully it becomes the main priority here soon.
Awesome. Well, Josh, thank you so much for spending your morning with us. This was great. And if I get any other questions in, I will make sure to chase you down and get them to you.
Sounds good.
All right. Have a good morning. And thank you everyone
for
tuning in.
You too.
Bye.